to speaking about the other branch of the peripheral nervous system. So thinking back to our general schema that we drew, where we have the two efferent branches, we've got the autonomic, which is further divided into sympathetic and parasympathetic. And then we also had this voluntary branch, right? This was called the somatic nervous system. And this is the one that controls our skeletal muscle. So this is the one that we have voluntary control over. We can't control our heart rate for the most part, unless we do so mindfully. We can't control our respiratory rate, glandular secretions, but we can physically make decisions about our skeletal muscle, and this is controlled by the somatic nervous system. So our first objective is to recognize the functional anatomy here, and then how the neuromuscular junction is organized, and really importantly, how the neuromuscular junction differs from the neuroeffector junction. So thinking about some of the contrasting anatomy, and I'm really going to be drawing analogies to what we said in the autonomic nervous system. So it's not enough to just study this in a silo. It's really important to draw distinctions and to be able to identify areas where the anatomy here, the function here, differs from the autonomic nervous system. So I'll continue to reference what we've already said about the autonomic nervous system. So the neurons here are not preganglionic or postganglionic. The neurons here are entirely differently named. They are motor neurons. Okay, they originate from a different area of the spinal cord, whereas we spoke about the autonomic fibers arising from the lateral horn, the somatic fibers, right, the motor neurons arise from the ventral horn of the spinal cord. So the anterior portion of this cross section of our spinal cord. The neurotransmitter involved here is only one type. So there's no epinephrine, no norepinephrine, no adrenergic receptors. There are no metabotropic or slow receptors. There's one type of neurotransmitter, that's acetylcholine, and there is one type of receptor that is a fast receptor. In other words, a nicotinic cholinergic receptor. In other words, an ionotropic receptor. Okay, so this is a little bit more simpler than what we saw in the autonomic nervous system. One receptor, one neuron. So there aren't two neurons the way that we see in the autonomic system. There's only one. It's called a motor neuron. There's only one neurotransmitter, it's acetylcholine, and there's only one type of receptor. It is a nicotinic cholinergic or a fast receptor. Okay.
I have a question about the hold on a second. On the invertebrate, what are the branches coming off of the spinal cord? Are those bone, or is that or is that like cartilage?
So here's the spinal cord. Coming off are the fibers that are going towards the spinal nerve.
Okay, so it's fiber.
Yeah, so these are the fibers. Okay. Yeah, so this is that ventral root, this is that dorsal root. And remember, we said that the root is different from the horn. So fibers are all exiting the spinal cord via the ventral root, but they're originating from different horns. So the lateral horn, right? Yeah, lateral horn is where we have the autonomic fibers. They use the ventral root to exit. Whereas here we're seeing that the ventral horn is where those fibers are coming from, also using the ventral root to exit.
Okay. Okay. Thank you.
Sure. Yeah. And so they're going to enter that spinal nerve, which we said was a mixed nerve. So there's got a lot of different fibers that are traveling back and forth, but they're in that single structure. Okay. Moving on here, we've got a motor unit. So a motor unit is described as one single motor neuron plus all of the different fibers that it is innervating. And we'll talk much more about the structure of muscle in chapter 12, but essentially one muscle group, let's say I take my biceps, for example, that is consisting of several different muscle fibers. And a muscle fiber is synonymous with a muscle cell. It's like the individual unit of muscle. And so we can have many fibers being innervated by a single neuron, okay? On the other end of that, a single fiber can only be innervated by one motor neuron, all right? So I'll say that again here. One motor neuron can innervate many fibers, but a single fiber can only be receiving input from a single motor neuron, all right? So if we're looking at this connection or this description of a motor neuron, it is a motor unit, excuse me, it is the single motor neuron that's coming out from the spinal cord here, traveling via this spinal nerve. It can branch into many endings that are going to different fibers. But if we look at a single fiber, like take this red fiber, for example, it can only be receiving input from one specific motor neuron. So in this instance, it's receiving input from motor neuron 2, right? So one neuron, many fibers, one fiber, one neuron. Let's speak now about what's happening at the neuromuscular junction. And this is sort of relying or calling upon what we described before in terms of those EPSPs and IPSPs. And these are essentially those graded potentials that are coming in, right, at different spots on the soma here. So we have the summation of these different potentials coming in on the soma, and we basically decide whether or not we have enough of an input. And the summation here is a little bit different, right, because we don't have inhibitory potentials. We don't have inhibitory events, or we don't have graded events happening here as such. What we see is an end plate potential, okay? And that is sort of synonymous with an action potential, but it is always excitatory. It's always an excitatory event. So let's kind of describe that here. So on the soma of our motor neuron, we've got an EPSP here, an IPSP there, and we're going to decide if we get to our threshold at the axon hillock. Okay, we're going to decide if this is enough of a membrane potential change to get us to a threshold here. When we get down to the end of this neuron, so although it's not being drawn or shown here, that action potential rips down the length of our neuron. When we get down to the end of that motor neuron, it can only be an excitatory ending. There is no inhibitory ending down there. There is no neurotransmitter that's going to cause inhibition. It's always going to be excitation of skeletal muscle that will result in muscle contraction, right? That is called an end plate potential. So just to kind of clarify that here again, whereas we can see IPSPs and EPSPs on the soma coming in here at the proximal part of this motor neuron, when we get down to the end of our motor neuron at the motor neuron ending at the axon terminal, we can only have excitation of skeletal muscle. Okay, so just going a little bit further here and adding some more detail to the events, we see synapsing between a motor neuron and a muscle fiber. The region of that synaptic communication is called the neuromuscular junction. And we have some unique terminology to the neuromuscular junction. So the axon terminal here is called the terminal bouton. So specific to the neuromuscular junction, we can refer to the axon terminal. In other words, that bulb-like ending of the motor neuron, we can refer to that as the terminal bouton. We can also refer to the region or the junction where we synapse onto the muscle as the motor end plate. So it's a highly specialized junction in that we see this tremendous amount of folding, which increases the surface area of the skeletal muscle on the adjacent side, and that is referred to as the motor end plate. Now, some of the differences that we see here, contrasting this to the autonomic nervous system, is all of the neurons are cholinergic. They all release acetylcholine, all of the motor neurons, that is. They are all cholinergic, all release acetylcholine. That is the only neurotransmitter that is encountered here in the somatic nervous system. They all bind to nicotinic cholinergic receptors. Okay, and nicotinic cholinergic receptors are these receptors here. We said that they were fast receptors. They allow for the flux of both sodium and potassium according to their ECDF. So we know sodium wants to go into the cell according to its ECDF. Potassium wants to go out of the cell according to its ECDF. And so when acetylcholine binds to this nicotinic cholinergic receptor, it's going to allow for the flux of both of those ions. And because we end up seeing more sodium come in than potassium leaving, this is always an excitatory event, right? So there's no inhibition of skeletal muscle. It is always depolarizing. It is always excitation of that motor neuron onto that skeletal muscle, okay? We also see these degrading enzymes. So these are also present because this is a cholinergic junction where acetylcholine is responsible for this transmission. We also see acetylcholinesterase, which is the degrading enzyme that helps to clear that junction. And the importance with acetylcholinesterase here is that it's really important to clear the junction in order to cease muscle contraction. We're going to talk about some toxins at the end of this. There are also some conditions where we don't see the clearing of this junction, and that actually brings about spastic muscle contraction. So muscles that are so stiff and rigorous and, in a constantly contracted state, which is due to the continuous signal that is being sent here because we have not cleared the junction of acetylcholine. So whereas in the autonomic nervous system, especially the parasympathetic side, it was important to clear the junction, it's even more important to clear the junction here, because then that would lead to continuous spastic muscle contraction, which can lead to fatality. If you think about things like the diaphragm, which is a skeletal muscle, that can become fatal if we have those spastic contractions of the diaphragm. Okay. All right, so we're going to review the events here across the neuromuscular junction. Much of this should look familiar. So I'm not going to go and draw this out, but we'll talk about some ways that there are differences. Again, just summarizing some of those main differences between the autonomic system and what we see here. So the events are, again, pretty synonymous with any synaptic communication. We have the action potential arriving at our terminal bouton. So again, the terminal bouton is the specialized name for the motor neuron ending here at the neuromuscular junction, whereas we've been otherwise referring to it as the axon terminal, so kind of synonymous structures. The voltage-gated calcium channels will open up. So these are also lining that terminal bouton. They're going to allow for calcium to enter our terminal bouton. And then these vesicles, which are holding acetylcholine here, are going to be released via exocytosis. They're going to dump across the cleft, and that acetylcholine can cross the junction and bind to the nicotinic cholinergic receptors. When they bind to the nicotinic cholinergic receptors, we said this can only result in an excitatory event. So it's going to allow for more sodium in than potassium leaving, which is going to result in a depolarization or an excitation of the motor end plate, okay? So this highly folded, specialized ending of the muscle here is called the motor end plate. And when we excitate or depolarize the motor end plate, that will then result in skeletal muscle contraction, okay? And so that is called an end plate potential. So the specific depolarizing event of skeletal muscle, whereas we would call it an action potential in any other excitable cell, in muscle, we call it an end plate potential, okay? After we've had that signal allowed to cross the junction and that synaptic communication take place, we then have to clear the junction via this acetylcholinesterase in order to cease that muscle contraction, in order to allow for the muscle to relax. So we do so via choline, which is a part of that acetylcholine, which can be uptaken back into the axon terminal here, the terminal bouton, recycled into a new vesicle, or we can degrade this into smaller parts and that can diffuse away, or it can diffuse away as is. Okay, so kind of similar mechanisms for clearing the junction as we talked about previously. This video is in Brightspace, so if you want kind of an easy 3D representation of this, make sure to watch this video to kind of summarize some of those events. Okay, a very great study tool here. This is a table that summarizes what's happening in the autonomic nervous system, what's happening in the parasympathetic division of that, what's happening in the sympathetic division of that, and then lastly, what's happening in the parasympathetic, not parasympathetic, in the somatic nervous system. So it's really important to be thinking about the main differences here. So the origin of these fibers. You also want to be thinking about the pathway, so how many neurons are involved. Whereas we look at 2 neurons in the autonomic nervous system, we have 1 continuous motor neuron here. And then lastly, you want to be thinking more importantly about the effector organ, right? So it's the smooth muscle, cardiac muscle in things like the autonomic nervous system, whereas in skeletal, in the somatic nervous system, it's going to be skeletal muscle, okay? And then some of the other differences that I won't go into, but you can summarize on your own, such as the neurotransmitter, such as the involuntary versus voluntary, etc. So an excellent study tool to summarize what's happening in the somatic nervous system and what's happening in the autonomic nervous system, both divisions. Okay. All right, so we'll take a pause here and look at a few of these checkpoint questions, and then we'll look at our remaining objective. So the first question here, which neurotransmitter is released by motor neurons at the neuromuscular junction, and to which type of receptor does this neurotransmitter bind? So this should be a lot more straightforward than the autonomic nervous system, because we only have one neuron and we've only got one neurotransmitter and we've only got one receptor. Excellent. So acetylcholine and nicotinic cholinergic receptors. Awesome. Acetylcholine, yes, that's correct. Nicotinic cholinergic receptors. So if you read on a question, somatic nervous system, epinephrine, automatically wrong, right? So there's no adrenergic receptors, no epinephrine, none of that stuff, okay? There are also no metabotropic receptors. So the receptors here are all fast in that they all result in an immediate depolarization of the motor end plate, okay? And that kind of makes sense, right? And we talked about this idea before, kind of the reasoning behind why we don't see metabotropic receptors at the autonomic ganglia, and also why we don't see metabotropic receptors in the skeletal muscle. We need this response to be nimble. We need it to be quick. If I'm thinking about moving my hands, if I'm thinking about taking a deep breath, moving my respiratory muscles, moving my legs, I need that to be an immediate response, a fast response. I don't need for there to be, second messengers in the way, or, amplifier enzymes or anything like that. It needs to be a quick response. It needs to always be depolarizing or exciting my muscle. And then in order to relax the muscle, I simply clear the junction. So it needs to be more of an immediate, fast response, and hence we don't have metabotropic receptors here. And then the last question here, how many motor neurons innervate a single skeletal muscle fiber? So how many motor neurons can innervate a single fiber?
1.
Excellent, thank you. So 1. So only one motor neuron can innervate one fiber. If I were to ask how many fibers can be innervated by a single neuron, that would be many, right? So one neuron, many fibers, one fiber, one neuron.